Good morning, class 10 students. I welcome you all on this online platform provided by JAGRITI classes. We were doing chapter 9th of class 10th, that is Heredity and Evolution. So we have already started with this chapter. I have gave you introduction and definition of these important terms. But now we'll be doing the further topics of this chapter. So in the last lecture, I told you the last topic was alleles. So what are alleles? Alleles are usually the different forms of a same gene. That is, each gene, I explained you, each gene is responsible for one character. We took eye color for an example, that is, gene responsible for eye color. The eye color can have different variation. In one parent, the eye color may be blue. In the next parent, the eye color may be brown. So the gene responsible for the blue eye color in one parent, if I again revise you with this, then you have different eye colors. So we have two parents, parent 1 and parent 2. Let's suppose for the first parent, you have the blue eye color and the next parent has a brown eye color. Now we have to predict, if we have to predict the eye color in the progeny or in the child, then these would be according to Mendel's laws. We saw Gregor John Mendel, Sir Gregor Mendel was a very, is known as father of genetics or father of inheritance because he proposed many useful laws which govern the phenomenon or the process of inheritance. So here we have that this gene which is responsible for blue eye color, this would not contain a single character B, but it has two forms, okay? So the parents have two forms of one single gene, and these two forms, these are known as alleles. So alleles are different forms or the two different forms or features of one single gene. These are known as alleles. And if we say for the brown, if I write, so the parent second having a brown eye color would be having two different forms of a gene or two forms for this eye color. They would be represented by letter small b, and these two small b here in turn are known as the alleles. So this topic we discussed with an example, with the diagram I showed you how the transfer occurs. Okay, what do you mean by the term alleles actually? We have discussed this. Now we are moving further and now we'll be discussing some important terms. In the previous video, I told you what do you mean by dominant trait and what do you mean by recessive. Then you can include one more point, that the trait which is dominant. Dominant means the trait which gets expressed in the future generation or in the offspring. It is shown, it is represented by a capital letter, while the recessive trait is written by the same small letter. Let's suppose if I am writing that, or what I have written here, if I am writing that this blue color is dominant. Dominant means it would get expressed in the child. So this trait, because it is dominant, it can be written by using capital symbols, and these symbols can be derived from the first letter of the characteristic feature. That is, blue can be represented using capital B and capital B. Whereas this brown, if we consider that the brown is the recessive trait, then whenever you are using the symbol capital, whichever symbol you are using in capital form for the dominant, the same symbol would be used for the recessive trait but in the smaller alphabet. That is, here for this recessive trait, I'll be using small b and small b. Okay. Here you can get confused because both the traits start from B. Now taking one more example here. I told you yesterday in the previous video lecture that you have 7 contrasting features in the pea plant, and these 7 traits have their dominant and recessive form. For example, we discussed a trait that is seed shape of pea plant, and this seed shape has two features or two traits. One is dominant, one is recessive. So the pea plant can be of two different seed shape. They can be round or they can be wrinkled. Okay, you can have these two different kinds of seed shape in pea plants, round and wrinkled. So round one is the dominant trait. I told you you have to learn all the 7 contrasting characters with their dominant and recessive forms. So yesterday we discussed these characters with the flow chart, with the diagram, and I hope you will now learn it. So round was the dominant one, whereas wrinkled seed shape was recessive trait or recessive character. So now I am saying that for the dominant, you represent the dominant trait by using capital letters. So for the dominant, the first letter here is R. So for the dominant, I'll be writing capital R, not only a single time but 2 times, because you know the gene for this seed shape, that is gene responsible for the round seed shape, would have 2 different forms which are called alleles. So we write 2 times the letter capital R. And similarly now, what should be the alleles for the recessive trait wrinkled? Okay, you can say that we can write capital W, or we can write small w, anything can be written. But what is the concept? What is correct to write here? So we said that the dominant trait is represented by the first letter of the trait, whereas the recessive trait is represented by same letters as the dominant but in smaller form. So for wrinkled, the correct would be to write small r and small r. Similarly, you can take one more example. Let's suppose we have seed color, and you have 2 traits for seed color. It is yellow and it is green. I told you do not get confused between the seed color and the pod color. Okay, the color of pea pod. Color of pod is also green and yellow. But I told you a trick, that is GPYS is dominant. Okay, so here we can see that G and P. So here this green would be dominant and yellow would be recessive. But talking to the seed color, then we said the trick was GPYS. It means green pod is dominant, so green pod would be the dominant trait, and yellow seeds are dominant, that means yellow seed is a dominant color. So here yellow is dominant. So please do not get confused in the colors, because both the colors are same, green and yellow. Here also you have 2 colors, green and yellow. But here if you are considering the color of seed, the color of pea seed, then yellow is dominant. Whereas if you are considering the color of your pea pod, pod is the external covering, the outer covering of the peas. Inside you have the pea seeds, and this is known as the pea pod. So for the pod color you have the green to be dominant, whereas for the seeds you have yellow color to be the dominant color. So if I'll write here, then yellow, because it is dominant, then it would be represented by the first letter 2 times capital Y, and here the green would be then represented by the same letters in a smaller form, that is small y and small y. But talking in the next example for the pod color, this green, because it is dominant, the green which is the dominant trait, it is represented by capital G and capital G, as the first letter is capital. And now the yellow color, which here act as the recessive trait, okay, it would be represented by the same letters of the dominant used in small. So it would be small g and small g. So this is actually the standard rule followed for writing the letters or the alleles of the given trait. I hope you understood this. Now we are moving on to the next 2 terms, which are homozygous and heterozygous. So what do you mean by homozygous? Homo here means same, and zygous represent to the word zygote. Hetero here means different. So homozygous means it contains 2 similar alleles for a particular trait. You can remember that generally the parents, whatever you take, the parents are always homozygous. But the progeny, offspring, child, these are one and the same things. Okay, all these terms we discussed in our previous class. So here heterozygous, the parents are never heterozygous, whereas okay, the parents in the first generation are never heterozygous, but the next parents may be heterozygous. Then you can have the generation, that is F1 and F2 we'll be doing, you can say the progeny can be heterozygous or homozygous. So now let's see, homozygous, it contains two similar alleles, two same alleles are present. The next is, in heterozygous, it contains two different alleles. That is, if I take a tall plant, the allele for tall plant can be capital T and capital T, or it can be capital T and small t, because you know, if I tell you that tall is a dominant character, so capital T would also give me a tall plant. Both the alleles are dominant here. Here in this case what you have, this capital T is a dominant allele, while the small t means shorter height plant. So this is a recessive allele. But because we know that when a dominant allele is present, either with the recessive or with the dominant, always the dominant allele gets expressed. That is, the plant which you will have will remain tall. So we can say that tall character can be represented by two cases. That is, either you write capital T, or you can write capital T and small t. What would be homozygous? The first one, capital T is called homozygous because it contains same alleles. So we wrote that when two same alleles are present, it is known as homozygous. So the fourth point you can include here as example, when you have both the alleles same, it is homozygous. So capital T is homozygous. For short plant, small t would be homozygous. Whereas if I talk about the example of heterozygous, then example would be different alleles. That is, one capital and the next one small. So heterozygous means presence of two different alleles for a single trait. The trait here which we are using is height of the plant. So particular trait, the trait which we used was height of the plant, and this height had two similar alleles, which is capital T. Whereas in heterozygous condition, the trait is again the height of plant, but the different alleles are what? Capital T and small t. Then moving on to the next, the genotype for homozygous possess either dominant or recessive, but never both the alleles. Okay, here also an example is given, and in our example we can see that genotype means the alleles. So the alleles may be dominant. It is written that both the alleles should be dominant or both the alleles should be recessive, but never both dominant and recessive alleles are present in the same manner. But in heterozygous, what we have, the genotype for heterozygous has both dominant and recessive alleles. Heterozygous condition means in the genotype you have the dominant allele and you have the recessive allele. Whereas in homozygous you have same allele. Either both the alleles will be dominant or both the alleles will be recessive. You cannot have a mixture of both the alleles, that is dominant and recessive would not be present. If dominant and recessive are present, then they would be heterozygous. The next is, it produces only one type of gamete, and this produces two kinds of gametes. Let's suppose, for explaining this, we have parents, and from these parents you have the formation of gametes. Okay, so we say we have two parents, P1 and parent 2, that is P2. Now from these parents, let's suppose the first parent has the genes alleles capital T, and the next has the alleles small t capital T. So can you tell me which parent is in homozygous condition and which one has a heterozygous condition? Parent 1 has which kind of condition? Homozygous or heterozygous? Correct. Parent 1, because it is having same alleles, Both the alleles are same, and when you have same alleles, okay, you call it to be homozygous. So parent 1 is homozygous. Now when the gametes will be formed, then you will have the formation of only one type of gametes. That is, we say all the gametes will have the allele capital T. So we are writing it produces only one type of gamete. Now can you answer me the gametes formed by parent 2? Which gametes will be formed by the second parent? Either there would be only one gamete formed, or there would be two gametes. There would be a formation of two types of gametes. Okay, that is, some gametes will have the allele capital T and the next gametes will have the allele small t. Because when we will be dealing with the laws of inheritance, I'll tell you that there is a law called as law of separation or law of segregation. It means that from the parents, whenever the gamete is formed, during the formation of the gametes, both the alleles get separated, so that each gamete either receives a capital T or either it can have a small t. But each gamete does not have both the forms. Either it can have only one form. So here we say that heterozygous produces two kinds of gametes. One is with capital T, one is with small t. This is an example. You have two different kinds of gametes produced in heterozygous. So I hope this is clear, the difference between homozygous and heterozygous. Now we have done with alleles, we have done with dominant recessive trait, homozygous, heterozygous. So now only one thing which you should know before we'll start with the cross, that is monohybrid and dihybrid cross. There are two words, phenotype and genotype. Okay, phenotype simply means the physical appearance, and genotype means the genetic constitution. So these are the difference. Phenotype means if I tell you that you have a tall plant, okay, so this appearance, that is, in appearance, whenever you see a plant, it appears to be tall. So this tall character would be the phenotype of that plant. But talking about the genotype, that is the genetic constitution. What kind of genes does the plant have? What kind of alleles are present in this plant? This gives you the answer of genotype. We already discussed that for tall plant, either you can have the alleles capital T, or you can have the alleles capital T and small t. So here we know that capital T means dominant trait, and here when both the dominant trait are present, the plant would be tall. But here when you have the heterozygous condition, heterozygous means different alleles in a same form. So when you have different alleles, then also because capital T is dominant and small t is recessive, so the dominant allele, capital T, will express itself. And when capital T will express itself, then the plant would be tall. Because when the plant would be short, can you tell me the genotype of the plant, that is the allele, the genes of the plant which is short? Try it out. Okay, the plant which is short, then it would have the presence of the recessive alleles. So the genotype would be small t, because we know that the allele responsible for tallness is capital T. For tallness you have the allele capital T. For dwarfness or shortness, short or dwarf plant, if we say, we have the allele small t. So you have two different kinds of allele. T represents tallness and small t represents dwarfness. Now I am asking you, if I want to get a tall plant, this T for tallness is the dominant allele and this is recessive. So whenever we discussed, I told you always dominant allele gets expressed in the future generation. But what is one condition when the recessive allele will get expressed? The recessive allele can only get expressed when the dominant allele is absent. When you do not have any dominant allele, then the recessive allele shows its expression, that is, it gets expressed and you get a short plant. So I asked you how you will get the short plant. So short is a recessive character, a recessive trait. So for getting recessive trait, you should have no dominant trait, no dominant allele. So the genotype for this short would be small t and small t, because both the traits, small t and small t, represent the recessive features, the recessive traits. And here we do not have any kind of dominant trait. So we say that when you do not have any kind of dominant trait, or you can say simply that when only recessive traits are present, then the recessive character, which is shortness or dwarfness, will get expressed. So this is the case of dominant and recessive, and I told you phenotype and genotype. If I tell you what is the phenotype, okay, tell me the phenotype and genotype for capital T and small t. So you will say for capital T and small t, the genotype I have given you. This is the genetic composition, T and T, capital T, small t, and the phenotype would be what? Tall. Because we know capital T is dominant, small t is recessive. Capital T will get expressed and the physical appearance of the plant would be taller. And if I tell you, can you tell me the genotype of short? So you should be easily able to tell this, that the genotype would be small t and small t. So this is the meaning of phenotype and genotype. Phenotype is the physical appearance of the plant, for example, tall, short, or you can say the round seeds, or you can say wrinkled seeds, anything. And if you are asked about the genotype, then the genotype for tall can be capital T, or capital T, small t. The genotype for short would be small t and small t. The genotype for round seeds, round is the dominant trait. So it would be capital R, or capital R with one small r. And the genotype for wrinkled would be what? Small r. So these words are the physical appearance, that is the phenotype, and these alleles or the genetic composition gives you the genotype. And now when we'll study the crosses, then you have to provide the ratio of phenotype, which is called phenotypic ratio, and then you have to write the genotypic ratio as well. So now we'll be starting with the cross. Cross is what? Cross means whenever you take two different kinds of parents and then you cross them, or then you multiply their gametes. From the parents we have the formation of gametes, then after the formation of gametes we'll have the progeny or the offspring, and then we'll observe that what is the phenotypic character of the offspring and what is the genotype character or the genotypic ratio of that offspring. Okay, so now moving on to the crosses. So you have two different kinds of cross which you have to study. The first is monohybrid cross and the next is dihybrid cross. So we have to study the two different kinds of cross, monohybrid and dihybrid. Firstly, we'll be seeing what do you mean by the term monohybrid and dihybrid. So it is written, okay, I'll write it in simple language for you to understand. Monohybrid cross. First we'll be discussing what do you mean by cross. Cross is what? When you take two parents, parent 1 and parent 2, then you have the formation of gametes from both the parents, then these gametes combine together. Okay, the gametes from both the parents, they combine together. Let's suppose the gametes are G1 and G2. So now the gametes will combine together, they will fuse together and a zygote will be formed. Here this zygote which is formed is actually known as the F1 generation. F1 generation is first, F1 means first filial generation. First filial generation is formed, and after the formation of first filial generation, we take the F1 generation and then we self-multiply or self-pollinate, or you can write self-cross. So we are taking the F1 generation next. So we take F1 and F1 and we cross the F1 generation together. So it is known as self-cross. Because F1 is crossed with F1 itself. So it is called self-cross. And then after this cross, you obtain a generation which is called F2. So this is the mechanism or the process of cross. This is meaning of the word cross actually. Cross means what you take is, you take two parents, parent 1 and parent 2. Then you need to form the gametes from the parents. So the gametes would be G1 and G2. Then what we did is, we cross the gametes together. Cross means to fuse the gametes in order to form zygote, we know. But this zygote actually here is known as F1 generation. The full form of F1 is first filial generation. Now this F1 generation or first filial generation, which is formed by the combination or combining of both these gametes, the gamete first and gametes 2. Now this F1 generation is crossed with itself. That is, you take two F1 species and you cross them out. So whenever this cross, because it is occurring between the same F1 species, it is known as self-crossing or self-cross. And then when you cross the F1 generation, you obtain F2 generation. This is known as second filial generation. So this is the basic mechanism of cross, and in this manner we will be studying the different kinds of crosses. You have to study two kinds of crosses, monohybrid and dihybrid. Now after this, that is after understanding the terminology cross, we will be studying what do you mean by the terms monohybrid and dihybrid. Monohybrid cross, you can say it is a cross involving only one kind of character. Okay? When only one character is involved, you can call it monohybrid cross. And in dihybrid, as the name suggests, hybrid means character, di means two. So when you cross out, dihybrid means the cross carried out between two characters. For example, if I say that I am taking a tall plant and a short plant, and if I do the cross between the tall and short, now in this cross what I am considering? The basis of this cross is height of plants. So if I am taking only a single character, that is height of plant, then it would be the monohybrid cross. But if I take two characters, that is, if I take, firstly I'll write it down here, the example, that is, if you take the height of plant and this is only a single character, then you will call the cross to be monohybrid. But for dihybrid cross, you need to take any two contrasting, any two characters. So let's suppose I am taking seed color and seed shape. So if I take both the characters simultaneously in a single cross, in one cross if I take two characters, then it would be known as dihybrid cross. For your syllabus, you have to study both monohybrid and dihybrid crosses. Okay? In fact, in further studies, you can also study trihybrid cross. But here we are limited with our syllabus of monohybrid and dihybrid. And you have to practice these crosses. Once you get these, you should know their you should know their phenotype and genotype ratio, and that's it. You have to do the cross, and then you have to write its phenotypic and genotypic ratio of both the generation of F1 and of F2. So let's see the points once. A monohybrid cross is a genetic cross between homozygous individuals with different alleles for a single gene. Okay, now we'll not be going into detail because you can be asked only the definition of the monohybrid and dihybrid cross with an example. And the main basis is how you have to perform the cross, and then you have to know the F1 and F2 phenotypic and genotypic ratio. So this is only our purpose. Just you can read the points out, okay, and we'll now be moving on to the monohybrid cross topic. So let's start with the first type of cross, that is monohybrid cross. Okay, So this picture which is in front of you tells you about monohybrid cross. I told you the general steps of a cross. First you have to take the parents and then we'll form the gametes from the parents. So first step is parents, then you have to make the gametes. The third step would be what? Fusion of the gametes to form F1 generation, and then the fourth step is fusion of F1 generation to form F2 generation. So these are the steps. You can see this is a monohybrid cross. Why? Because you have yellow and green colored seeds, and it is a monohybrid cross because it involves only one character. And which character does it involve? It involves the character of seed color. And hence, because we are only studying the cross between individuals based on seed color, it is called as monohybrid cross. So in monohybrid cross, it is involving a single character seed color. Now you have the parents. P here represent what? Parents. So you have two parents. The first parent has a yellow seed color and the next has a green seed color. Okay? Now see, you have two parents, parent 1 and parent 2. The first parent has a yellow seed color and the next has a green seed color. And these all crosses are the experiments done by Mendel, Sir Mendel, on pea plants. So all the characters, the examples which you will take in the crosses should be from the characters of the pea plant. So if you remember that you have two different seed colors of the pea plant, one is yellow and one is green, and I already told you a trick to learn the seed color and pod color, and the trick was GPYS. So for the seed color you have yellow color to be dominant. It states that green pod color is dominant, yellow seed color is dominant. And because we are considering seed color, now you should know two different types of seed color what you have. Then you should know among the two which is dominant, which is recessive. So we know that this yellow color is dominant and the green color is recessive. Okay? After knowing the dominant and recessive, you have to form the alleles or the genes. So for parents, I told you the parents which are the initially taken as parents, they are always homozygous. Homozygous means they will have similar type of alleles. So for the yellow color, because yellow is a dominant trait, I would be using the first letter in capital for the dominant trait. So it would give me capital Y and capital Y. I hope you understood this. We have discussed it, okay, in the previous section. And here the green is recessive. So for the recessive trait, we use the same alleles, the same alphabet of dominant, but in the smaller alphabet manner. So for green, you have two alleles, small y and small y, and for dominant yellow color, you have capital Y, capital Y. So hence in the picture it is given, the parents are capital Y, which means yellow, and small y, which means green. So these, now what you have to know, you have decided the alleles of the parents. Now the next step after parents is the formation of gametes. So you see, I told you from the homozygous parents, these parents are what? Homozygous. Why we are calling them homozygous? Because they have same type of allele. Parent 1 has capital Y, parent 2 has small y. Because both these alleles are similar, such parents are called as homozygous parents. Now we need to form the gametes from these parents. So what would be the gametes? I told you from the homozygous parent, only one gamete, only same kind of gamete is formed. That is, all the gametes of this parent would have the allele capital Y, and all the gametes of this parent second would have the allele small y. So the gametes would be capital Y and small y. And the gamete only receive a single copy of the parent's allele. That is, when you have two alleles, the gamete will only contain one. Okay, now moving on to the next. After the formation, see here also the gametes are formed same. It is written each homozygous parent. I hope now you understand the term homozygous, okay? Each homozygous parent in the parent generation produces only one kind of gamete. Why? We have already seen from the homozygous only one kind of gamete is formed, and if you have heterozygous parent, let's suppose if you had capital Y and small y as parent here, then you will have the formation of two gametes. Some of the gametes will receive the allele capital Y, and next type of gamete will receive the allele small y. So we say that if you have heterozygous alleles, then you have two types of gamete. But if you have homozygous alleles, that is the same alleles, then only one kind of gamete is formed. I hope now the concept is well clear. Now what you have to do after the formation of gametes, you have to combine or fuse the gametes together, and when you will combine the gametes, we will be getting F1 generation. Okay, because of this mechanism only I told you the basic steps of the cross in the previous slide. What are the basic steps? First you have the parents, then you have to form the gametes, and then you have to combine the gametes to get the F1 generation. So here when I combine the gametes, I get capital Y and small y, which is in fact the F1 generation. So you can see the F1 generation is capital Y, small y. And now this generation, is it homozygous or heterozygous? Correct. It is heterozygous because it contains different alleles. One is capital, the other is small. And hence, the heterozygous F1 offspring produces again two types of gametes. Now the next step is this F1 generation which we got is heterozygous. Okay. Now we will be carrying out the next step. What is the next step? From the F1 generation, you have to form the gametes. So you see you have a heterozygous F1 generation. So the gametes formed would be of two types, because from heterozygous you have two different kinds of gametes, I told you here. So one gamete would be capital Y, other gamete would be small y. So these would be the gametes of F1 generation. And I told you that after obtaining F1 generation, what you have to do? The F1 generation which you obtained here was capital Y, small y. Now after F1, we have to take the two F1 generation and you have to self-cross them. So now I am writing the two times, this is my first F1, this is the second F1. Now for self-crossing, you have to form the gametes from the F1. So the gametes from this F1 would be capital Y, small y. And you will have same gametes from the next capital Y and small y. So this is your first parent, that is F1, and the next is again also F1. So now what we'll do is, we'll make a square. To show the cross, you make a square. In the first column, we are writing, this is my first F1, and here I am taking the second F1. That is, this is parent 1 again and this is parent 2. Now from parent 1 I got the gametes capital Y and small y. So write the gametes of F1 here. Okay, and then the next F1 which is your parent 2. You have two different kinds of gametes formed from parent 2. From parent 2 you have the gametes capital Y and small y. Now what you have to do, you have to cross them. That is, in the first block you have capital Y, capital Y. In the next you have capital Y and small y. Similarly, if you cross them in this block, what would come? Capital Y, small y. And in the last it would be small y, small y. So these are the four types of different generations which you will be getting for your F1 or for the monohybrid cross, and these generation would be called as F2 generation. Now you have to find the phenotype and genotypic ratio. Phenotypic ratio and the genotypic ratio of F1 and F2. So talking about the phenotypic ratio of F1. So let's move on to the F1 generation. This whole is your F2 generation. So in the F1, what you got is, see, it is also given here, okay. So in the F1, the phenotypic ratio were 1:1 and the genotype was Y and Y. So for F1, there is no need to find the ratio for F1. Okay, you can directly find the phenotypic and genotypic ratio for the generation F2. So now you can see in the F2 generation, you have 4 types of offsprings. The first offspring, that is capital Y, then you have capital Y, small y, then you again have capital Y, small y, and the last is small y, small y. We know that capital Y, that is yellow color, is dominant. So here it would be what? Which color would it show? This would show yellow color because you have both the dominant alleles. Here you have one dominant, one recessive. But always whenever the dominant allele is present, it expresses itself. So here also the color would be yellow. Here also the color would be yellow. But here you do not have any dominant allele. So in the absence of dominant allele, the recessive allele gets expressed and the color would be what? It would not be yellow. Small y means the color green. So you have phenotypic means what is the physical appearance. So you have 3 plants as yellow and 1 plant to be green in seed color. So the phenotypic would be yellow is to green. We are finding the phenotypic ratio. So the ratio is 3:1. Okay, this is the ratio for F2. And talking about the genotype, genotypic ratio means what? The genetic constitution. So for the genotype, how many different kinds of genotype you have? You have capital Y, then you have capital Y, small y, and then the next type is small y, small y. So these are the 3 different types of genotypes available for the F2 generation. See, you have 3 kinds of genotypes. Capital Y is present only 1 time. Capital Y, small y is present 2 times. So you will be writing 2 and then small y is present only a single time. So this would give you the genotypic ratio and the phenotypic ratio of F2 generation of which cross? Monohybrid cross. Now these ratio for the monohybrid cross remain same. Whatever character you are taking, the ratio would be 3:1 and 1:2:1. Now you can practice other crosses. This one is dihybrid in fact. And now you can practice this cross, that is, you practice on your own, you take the character height, then you take the parents tall and dwarf, the gametes would be capital T, small t. When you cross the gametes, you would get this as the F1 generation. Now we are mating, which means self-crossing the F1. F1 was capital T, small t. When you self-cross, you will have 2 different types of gametes, then you can form a square. Similarly, which we formed here, that is capital T, small t, capital T, small t. This is your parent 1 of F1 and this is your parent 2 of F2. And when you cross, you will get the combination. This combination you will get. And now you can find the phenotypic and genotypic ratio of the F2 generation. This table would give you the F2 generation. Okay, by the crossing of 2 F1, you get the F2 generation. And the phenotypic ratio would be tall is to dwarf would be 3:1. And the genotypic would be 1:2:1. So you can say that these are the similar ratio which we obtained. Okay. Here also we got the phenotypic 3:1 and genotype 1:2:1. And similarly, in this example also you are getting the same ratio. So you can say that we can learn the ratios of the monohybrid cross of F2 generation. The phenotypic ratio of F2 generation is always 3:1. 3 is for the dominant trait, 1 is for the recessive trait. And here you have, this is for the pure dominant allele, this one is for the, that is 1:2:1. This ratio is for pure dominant. Pure means homozygous dominant. This is for homozygous recessive, and this too is for heterozygous, that is mixed and dominant. Here dominant is tall, recessive is dwarf. So you have to form pure means homozygous. So here it would be capital T, capital T. This would be capital T, small t. Dominant is tall, hetero means both the alleles are present. And homozygous recessive, it means small t, small t. So you can learn the ratios in this manner, okay? So this completes your monohybrid cross. Do practice a lot of examples given in your books too, and do practice the cross. Without practice, you will not be able to do these kind of crosses. It would require practice at the initial stage. And then you will learn the ratios, and then you will find it very easy and very good to do such kind of crosses. In the next class, we will be discussing the dihybrid cross. So thank you for joining today's class. If you have any kinds of doubt, any kind of doubts, you can contact us anytime. I hope you understood the cross and the introductory video which we discussed. Mendel, who was Mendel, the 7 contrasting characters of the pea plant. What were the reasons why Sir Mendel chose pea plant? Okay, everything we discussed, and this was again an important lecture. In the next lecture, we will be starting the dihybrid cross, which is this one. So we'll be starting with this. This is dihybrid cross, where you take 2 different kinds of characters. See, here we took the yellow and the seed shape and seed color. So here we are taking 2 characters, seed shape and seed color. And because I told you in the definition, when 2 characters are involved in the cross, you call it to be a dihybrid cross. So we'll be discussing with this cross in the next future class. So thank you for joining the class, and I hope to see you very soon.